Very good morning to one and all, respected professors and my dear participants. We heartily welcome you to the second day of this webinar on principles and practices of phytochemical research. Now it's my pleasure to invite Dr. Xavier Rajaratnam, HOD Chemistry, Department of Chemistry, Sacred Heart College, to invite the participants. Over to you, sir.
Thank you, Dr. Collins. I have a pleasure in inviting Dr. Jancy Mary, the HOD of Chemistry from Auxilium College. Over to Jancy, madam.
Respected Reverend Dr. John Alexander, Secretary, Reverend Dr. D. Maria Anthony Raj, Principal, Reverend Dr. K. A. Maria Aroki Raj, Additional Principal, Shift 2, Reverend Dr. Praveen Peter, Vice Principal and Controller of Examinations, Reverend Dr. D. Theophil Anand, Vice Principal, Sacred Heart College, Reverend Sister Alice Katie, Secretary, and Dr. Sister R. Jayashanti, Principal, Auxilium College, Reverend Dr. S. Innasi Muthu, Director, Xavier Research Foundation, St. Xavier's College, Autonomous, Palayangotte, Tirunelveli, Dr. S. Xavier Rajaratnam, Head Department of Chemistry, Sacred Heart College, members of the organizing committee, staff, and my dear participants. A very happy and pleasant morning to all of you. I am happy to say a few words of felicitation on the second day of the webinar on principles and practices of phytochemical research. My hearty congratulations and appreciation to the Departments of Chemistry, Sacred Heart College and Auxilium College for organizing the seminar on phytochemical research. Medicinal plants are nature's gifts to humanity and play a vital role to safeguard our health. India is one of the most medico-culturally diverse countries in the world, where the medicinal plant sector is part of a time-honored tradition that is respected even today. India is well known for its rich traditional systems of medicine, that is Siddha, Ayurveda, Unani, Amchi, besides a vast reservoir of living traditions in ethnomedicine. Ethnomedicine is as old as man's history. Ethnomedicine examines and translates health-related knowledge and theories that people inherit and learn by living in a culture. Each society has a particular medical culture or ethnomedicine, which forms the culture's common sense or logic on medicine. I think somebody has to mute your audio. The World Health Organization estimates that 80% of the people in developing countries rely on traditional medicines, mostly plant-derived drugs for their primary health needs. In the existing scenario, where we are waiting desperately for medicines or vaccines for the dreadful COVID-19, and when we are advised to take plant-based remedies, this seminar would be an eye-opener for us to understand the importance of research on plants and plant products. Father Innasi Muthu is synonymous with ethnomedicine, as whenever I hear his name, only his research on medicinal plants in specific areas and studies on their biological and medicinal activities comes to my mind. I'm sure we are going to delve into the field of ethnomedicine in the next 1 hour. I wish the organizers and all the participants a very fruitful session. Thank you.
Thank you, madam. Thank you for our words of presentation and introduction. Pleasure to invite Reverend Dr. Praveen Peter, the Vice Principal and Controller of Examination of Sacred Heart College, to introduce the resource person, Dr. Innasi Muthu. Over to Father Praveen.
Good morning to all present here. I take it a great privilege to introduce Reverend Dr. Ignacy Muthu, who is going to be the main speaker of this session. Reverend Dr. S. Innasi Muthu is a Jesuit priest belonging to an international religious congregation, the Society of Jesus. He was studious right from his younger days. He acquired second rank, university second rank in BSc Botany and stood first in MSc Botany in the university. He completed his MPhil in 1983 and PhD in 1985 in the field of genetics from the University of Delhi with outstanding merits, and his DC in the field of plant biotechnology from the University of Madras in 2001. He is one of the very few scientists to acquire this degree. More than that, he has served in prestigious colleges in various capacities, like hostel warden, vice principal, principals, directors, etc. In October 2000, he was appointed Vice Chancellor of Bharatiyar University, Coimbatore. In June 2002, he was appointed Vice Chancellor of University of Madras. And presently, he is the Director of Xavier Research Foundation, St. Xavier's College, Palayamkotte. As Principal and Vice Chancellor, he made a mark in administration and academic reforms like introducing teacher evaluation by students, establishing poor students scholarship, introducing choice-based credit system, introducing semester system with credits, compulsory social service scheme, and transparency system of handing over the photocopy of all the answer sheets to the students. He has published more than 600 research papers and 70 books. I repeat, he has published more than 600 research papers and 70 books. His articles have been cited more than 22,000 times by other scientists. He has also contributed to enhance the value of students by giving value education books like Basic Good Manners, Values for Life, Being Happy and Successful, Skills and Qualities for Effective Life, and Shining in Life. Some of his books on biotechnology, bioinformatics, bioethics, and environment are used as textbooks in universities and colleges. His Tamil books on environmental awareness got the Best Book Award from the Tamil Nadu government, and he was also given the Kamarajar Award for research on environmental management by the government of Tamil Nadu in 2009. The Royal Etymological Society, London awarded FRES, Fellow of Royal Etymological Society, for his outstanding contribution in entomology. He is also the Fellow of National Academy of Agricultural Sciences, New Delhi. He is Emeritus Scientist of UGC, CSIR, and ICMR, New Delhi. He has carried out already more than 40 government-funded major research projects. He has many patents. Ponneem, an eco-friendly biopesticide, was developed by him. One insect species is named after him, Jactrips ignacimutu. One natural molecule is named after him, Ignaciomycin. He has also visited many countries such as USA, Japan, Germany, Switzerland, etc. The list goes on. We are indeed privileged to have him as our resource person for this first session. Thanks, dear Father, for consenting to enlighten us today by your inspiring presence and depth of knowledge.
Thank you.
Over to Reverend Dr. Inaximuthu.
Thank you. Thank you, dear friends, and greetings to all of you. I'm also immensely happy to share with you some of the knowledge that we have gained through some research along with my students and other faculty members. Also, I'm really congratulating both these departments for taking up this wonderful webinar so that other students and staff can benefit. And also a word of appreciation for the kind introduction that you all gave about me. I often say, what you are is God's gift to you. What you are is God's gift to us, and what you become is our gift to God and to the society. So all of us, by God's grace, of them and blossom and bring out something for the benefit of future generations of people, then I'm sure we are doing what we are able to do. Now, the topic in which I'm going to speak is related to the scientific validation of some herbs used in ethnomedicine. You are aware already, the introduction was given, how many, yeah, excuse me, I think, Tamil, internet, yeah, pardon? Internet வேலை செய்யலைன்னு நினைக்கிறேன் இல்ல?
Father, we can able to hear, Father. You can
Yeah, I'm just coming through. You are hearing me now?
Yes, Father. Yes,
Yeah,
Father.
yeah, okay. So we are just now opening the topic, scientific validation of some herbs used in ethnomedicine. As I already mentioned, I congratulate both the departments for organizing this and also the principal for the kind words about me. We are all God's children, and every one of us have some talents, and all of us can certainly bring out these so that others can benefit and grow. Now, you know very well, ethnomedicine is one of the ancient systems of medicine that our people have been practicing for a long time. And every plant has certain property. And if only we are able to do certain bioassay and confirm the claims of the people, then it is adding to new knowledge as well as also verification of what is being done. So what I'm going to share with you is related to some of the research that we have carried out for many years in our laboratory. Already the introduction has been given. Plants and plant-based medications have been the prime therapeutic tools for human beings all these years. New drugs are discovered in various ways, and the discovery of new biomolecules are traditionally used from medicinal plants. And one of the approach is to discover these traditionally used materials to develop into novel drugs. As India is one of the 12 mega biodiversity countries, we have excellent biological diversity as well as good traditional knowledge. So our contribution to global health market is comparatively less, and the need for scientific studies is a major concern. Compared to China, we are much behind. Here I am giving you the example of one of the scientists, Yu Tu, who was born in China in 1930 and also was aged 85 when she was given half of the Nobel Prize for discovery in medicine. That discovery was related to development of a drug from a plant, Artemisia annua. And this material, they were able to do for many years of research. Ultimately, they came out with the molecule, artemisinin, and later on they modified it synthetically, a semi-synthetic modification of the original molecule, which gave very good results to combat malarial parasite. And therefore, this was recognized as one of the good examples of traditional medicine leading into mainstream medicine. And ultimately, she was recognized for her hard work. And today, lots of recipes are available in our traditional system. If they are scientifically analyzed and validated, I'm sure we will be able to get many more novel drugs. The implication of this study to our Siddha preparation cannot be overemphasized. Now, documentation and validation of traditional claims in Tamil Nadu by a group, that is what I'm going to share with you. Traditionally, lots of plants have been used to manage. Many of these claims were not verified scientifically. To fulfill the scientific gap, we have been working on some plants. For example, here, Cassia fistula, this is the flower is used traditionally for treating diarrhea, skin disease, fever, even leprosy. The Tamil name is Sarafondrai. Now, we wanted to verify its activity, particularly because it is related to diarrhea. So we wanted to check, and what we did was do the customary chromatography, isolation and separation of these molecules. So we starting from the crude extract, we go to the column chromatography and we collect the different fractions, combine the fractions together and see the activity of these fractions, and ultimately come out to some useful fraction which is yielding a compound, and that compound we were able to identify as a Rhine compound, known compound, and it's an anthraquinone. So all the data related to analysis and so on is available, so which we can check. Now here we found some of the activities related to the bacteria, for example, were very close to the streptomycin positive control, for example, against Bacillus subtilis and Staphylococcus aureus. They were all good indicators to show that this traditional use was more or less confirmed through our scientific validation. And again, we checked also for antifungal activity and some of the fungi which were also useful, particularly here, for example, the results were close to the ketamine which we were using at positive control. So what is implied here is that any material that we come to know as a traditionally used material for a particular ailment, we can bring in the scientific validation to add more merit to what we are doing. The second one here is a Peltophorum spherocarpum flowers. It is in Tamil called Porkondrai. The bark is traditionally used to treat dysentery, and it is also externally used as lotion for eye trouble, muscular pain, and sores and things like that. So we wanted to verify again using the chemical methods of isolation from the crude extract, using different solvents and so on, and ultimately coming out which is the best crude extract that is active, and then go in for fractionation, and ultimately we were able to get a compound which
Further, this is Collins.
We could not hear.
Further, are you able to hear me?
Yeah. Are you able to listen to me? You hear me? Yeah?
Yes, Father.
Yeah. Okay, there was a current cut, so there was a little bit of disturbance.
No problem.
Okay, now, this burgeonin we were able to isolate using chromatographic techniques, and we wanted to verify its activity. Yeah, this burgeonin showed antibacterial activity, as we were able to also verify with the, yeah. So this is what we observe here, some of the antifungal activity related to burgeonin, and it was also very close to some of the standard materials, flucosanol, which we used. So in the standard comparatively also was showing good activity. The third one, which I'm sharing with you, is Tordelia asiatica leaves that traditionally used to control malaria, fever, stomachache, toothache, and so on. Sometimes even cough and nasal and bronchial pains. And here again, we followed the same methodology using column chromatography, collected various fractions, and went in for the isolation of compounds. Here we were able to get flindrosine, an alkaloid, and then eulopterol, coumarin, and these were used to check for their antibacterial activity. And here again, you can see how these were in comparison to the positive control, they were able to show some good activity against a few pathogens. Then we went into another plant called Phyranthus indicus, and this is also traditionally used to treat many venereal diseases. From here, we were able to isolate two different compounds, 7-hydroxyflavonoid and theophane A, and these were also checked for antifungal and antibacterial activity, and the zone of inhibition clearly indicates, compared to the positive control, we were able to get also some good results. And this is against fungi. So some of them are able to show important activities, showing that there is clear indication of how they can also be controlled. Then some viruses also we checked, and for some of the viruses they were shown of inhibition also good amount. Then the antibacterial activity of Nymphaea stellata. Nymphaea stellata is a plant you know very well, growing in many water bodies and so on. From the powder rhizome is given for dysentery, diarrhea, and hemorrhoids, and the decoction of the flower is usually given to treat diabetes. And in the seeds are used for stomachache and restorative action and so on. So from here we were able to identify ellagic acid using these chromatographic techniques, and again we were able to find out the activity for antibacterial and some clinical isolates of bacteria, and some of them compared to the positive control we were able to see some good results. And we also had some idea about antimicrobacterial activity of some Indian medicinal plants, because sometimes traditionally people use certain plants for pulmonary ailments. And so we isolated a compound from Solanum torvum and referred to as methyl caffeate. It slightly yellow crystals we were able to get, and this was also again showing good activity. Again, another plant we selected, and from Rheum imodi, one more imodine compound was isolated. Then from Arathoda vasica, we also isolated two different vasicin associate, then vasionoin and so on. And this also we were able to use for anti-tuberculosis activity. So these are some of the different compounds that we checked for anti-tuberculosis activity. And you can see here the number of names of the plants, the compound name that we were able to obtain, and the strains which we checked. And many of them were showing good activity at low concentration against tuberculosis bacterium, which is taken up by the Council of Scientific Industrial Research for further progress, and hopefully some modifications can be done using synthetic, semi-synthetic modification to find a good drug for tuberculosis. This is again the same continued. Some of these are immediately useful from the point of view of how they can be really taken up for further study. Now, the other area is where we have used for study was related to diabetes. For example, some plants for Nymphaea stellata are used for anti-diabetic study. So here we isolated from Nymphaea stellata a new compound called nymphaeol, and this nymphaeol is a new compound to science. So we really went for a patent for this also. Using the same chromatographic techniques, we were able to identify this molecule, and we found really a good kind of report, particularly when we did the immunohistochemical studies using the islets of Langerhans cells to see whether there was a rejuvenation of affected Langerhans cells. As you can see, the control, most of the cells were affected, but through our treatment, there was also an improvement and a reduction in the effect of the diabetes. So certainly this is validated through this scientific research. All these things have been published in form of papers, which you people can collect when you do a Google search. Then we went to another one traditionally used for diabetes, Caseria esculenta, another plant we removed using various solvents. Ultimately, we got 3-hydroxymethyl xylitol. This is again a new compound. We were able to also file a patent for this. And this again showed very good activity in regard to the islet salpingohans, where there was a rejuvenation of the cells based on the treatment with this. Then we went in for another plant, Phyla nodiflora, where this cytosterol is a very commonly available in different plants, but we were able to also identify and verify its activity. And this also showed some of the activities related. Then another extract also we were able to take from Agel marmelos. We got another molecule, dimethyl allyl sulfonitinol. And all these we were able to check using animal models to find out the expression of these things, particularly with regard to diabetes or obesity and things like that. And some of them had a very good effect related to these. And we could see that the expression even at the gene level. Then we also identified anti-hyperlipidemic compound. This is a compound which is very useful to reduce our weight, bulky weight, and obesity can be treated also. So some people use this even today, and we were able to identify a compound which is showing this activity. And we analyzed this with the help of animal model, and in 28 days it was able to reduce the adipose tissues which were stored in storing the fat. So this was also clear. And we also did the analysis with regard to another compound, and it is also showing clear anti-obesity activity. Then coming to anti-cancer activity, there are some plants which are already available. So Tinospora cardifolia is a plant where people have done some work, and we were able to identify a compound, epoxychloroidine diterpene. And this compound was again showing a good activity, particularly in controlling the growth of cancer cells in the normal liver, and then going into preventive and curative treatment. We were able to really see the nodules very, very small in the treated materials. Then anti-inflammatory molecules, or a lot of plants are there which are showing anti-inflammatory activity. So we were able to also identify some of the molecules related to Cleidodendron flamidis, for example. It is traditionally used to treat arthritis, and we were able to identify a compound, dihydroxy-2-methoxysodium butavanate, and we analyzed and checked whether it is showing real anti-inflammatory activity by the help of TNF expression. And you could see up to the gene level expression, protein level expression, we were able to confirm their activity as anti-inflammatory. Then another compound we were able to also identify, and it also showed good anti-inflammatory activity. Now, coming to another area, there is not much research happening in what is called non-alcoholic fatty liver disease, NAFLD we call it. So non-alcoholic fatty liver disease is one which is now spreading also quite a bit. Even though people do not drink alcohol, they seem to collect into fatty liver, and there is a possibility for giving some treatment. So we had many, many different kinds of molecules which we have isolated from plants, and some of them even semi-synthetic molecules. We analyzed them for their activity against this anti-NFLD, and many of them showed a very good activity, and we even went for ultrasound scan images, and then also the reduction and so on. And ultimately, we showed that they are also very good, useful molecules. And again, andrographylide derivatives of NFLD, they were also checked, and using various methods, bioassay methods, we were able to prove that andrographylide derivatives also have very good effect in reducing the effect of NAFLD. All this also, as I said, we have already published many of these papers, sorry, which you people can have a look at. Then you can see here in this picture how the effect of on the animal when it is given NFLD developed through this chemical treatment, and ultimately it is getting reduced because of the treatment which we give with the help of the plant molecule. Then we have also done a few analysis for microbial sources. Along with the plants, we also have done some work on using some microbial things like actinomycetes and so on. So there are quite many different types of materials, particularly Streptomyces species, which already have given a lot of antibiotics for our use. So we have been able to isolate many molecules. One of them is this new molecule that we named after Ignaciomycin, and then very others we were able to, they were showing good antibacterial, antifungal compounds from many of them. Then like, for example, another one is bleomycin. Bleomycin, again, we have gone for a patent. And these are some of the examples to show that there are also natural molecules that are available, which are antimicrobial, which are anti-cancer, from the actinomycetes and the bacteria that are around in our soil, which could easily be used for our study. Now, we isolated some of them, and using, as I said, the usual methodology of chromatography and so on, isolate these molecules, and we also check them not only for bioassay activity, we also check them for these molecular docking analysis, where the ligand is binding and what kind of energy level is being used. So making use of this, we also predict how a particular molecule will be able to bring out its mechanism of action and so on. Then we have done small work related to vector management, particularly using these plant and materials, compounds isolated and so on. Mosquito is a major problem, and as a vector, it is spreading so many diseases. So we have also identified some materials which are useful to control them. One, for example, is an oil a molecule from oil, Polygonum hydropiper. From there, we were able to identify a confertifolin compound, and this confertifolin was able to also control this mosquito spread. Then another compound we were able to, niloticin, isolate from another plant, which is also able to show good activity against mosquito. So in short, what I wanted to share with you is simply a glimpse of what we can do with the biodiversity available with us, particularly doing certain bioassays using the chemical methods that are available, and then isolating compounds and even modifying them later on, and verifying with certain bioassay that will certainly give very good results for us to move forward. We have a long and timely reviewed and updated tradition of medicinal plant usage in our country. Number of medicinal plants have been used in our tradition for handling various chronic ailments. Unfortunately, due to lack of scientific validation, it is a real major lacuna, and there is much less contribution towards this global health care. So it is high time for us to really go in for finding newer areas of discovering medicine from our natural resources, because they are healthy, they don't have side effects mostly, and they will be also very much useful for our benefit. So Interdisciplinary approach is needed to discover the potential of many of these plants for the demands of the global health care. Now, these are some of the patents that we have already obtained. I am just giving you a highlight about it. And we have also filed a lot of patents. This is work done by many of my students and scientists who have been associated with me for the last 20 years or 25 years. So I thank them also for whatever they have contributed. And ultimately, as our Albert Einstein, great scientist, told us, if we can't explain something simply, means we do not understand well enough. So let us hope that what I have explained to you gives you some input and some direction for future research, and hopefully it will benefit all of us. Thank you. God bless you. Yeah, Collins?
Yes, Father. Thank
Yeah.
you so much for
Yeah.
your wonderful lecture
Thank
on
you.
this ethnomedicine. Now the session is open for discussion. I request the participants to ask their questions either through the Zoom chat or through the YouTube chat. You can post your questions. Father, we have received one question
Yeah.
from a participant.
Yeah.
Sri Sasire Sai Kritika, she has asked, is there any criteria to choose the plants, or it can be chosen on a trial and error basis? That's a question you would like to ask.
Yeah, choose the plant. How do you choose the plant? First thing is, once you want to check a plant, go through literature search. Make a study whether any work has been done. If already a lot of work has happened, and it may not be useful to select that plant. So check the literature. Second one is whether that plant has been in use by people for many years or many traditions. Sometimes people choose some plants which are toxic, for example, which should not be taken up by human consumption and so on. So those plants we should avoid. So kindly check those which are needed, which are being in use by people, and which not much work has been done. Such plants can easily be chosen for any kind of analysis or bioasset study. Yeah.
Father, thank you so much. Hope
Thank you.
it would have
Thank you.
answered the questioner.
Similarly, further,
Yeah.
similarly, when you select a plant, how do you find the activity of the plant? You just go through every activity, or you just
Yeah,
the
yeah.
past history of the plant? How do you?
Knowing what kind of activity a plant has got, first thing, as I again told you, please check the literature already available. Lot of work has happened in a particular plant or particular molecule belonging to that plant. People may have already reported. If nothing has been reported, very easy for you. You start from the basic, what you are interested in. Suppose you are making a study on anti-cancer, then go in for checking using first in vitro cell lines, and then if some activity is shown in the in vitro cell lines by a particular compound or even extract, then go in for isolation of the compound from there, then go for fraction and compound analysis and things like that. So depending on the aim of your study and depending on the so-called area in which you are wanting to work, you can move forward. As I said, there are so many areas: antibacterial, antifungal, antiviral, then inflammatory, obesity, and anti-cancer, diabetes. Any number of areas are available where plants can be checked for their real activity. The most important thing is checking them in the literature and then moving forward to fill the gap. Okay, thank you.
Okay. Do we have to, or do we have to do the analysis by ourselves, or is there any institutes available for the activity study?
Every laboratory may not have all the facility that are needed to carry on from the start to the end.
Yes,
For
yes,
example, you take a raw material of the plant, and then at the end is to really do the bioassay and find the result. So to all these things cannot be really available in a particular place. So certainly we need to depend on a few other laboratories where people can help us, collaborate with other scientists who are working Naturally, that is needed also, no doubt about it. One example I'll tell you, many of us do not have, for example, NMR and all such kind facilities. So we go in for sophisticated instrumentation center to get those things. But we need to be interpreting it. So we need a scientist who has knowledge about it. So with the help of the scientist, we get the interpretation and also assign the molecule and then structure and so on. So certainly interdependence in research is very, very important. And I recommend that we try to collaborate with the already available scientists in different institutions to achieve what we are interested to achieve.
Okay, okay, Father. One more final. It is it essential to isolate the compound and check the activity, or we can take the crude extract and check all the activities?
No. We cannot really go for the final one. First, you must show that the crude had shown some activity. So from the crude, we moved into fractions, and fractions, this particular fraction showed some activity. Then the compound. In synthetic chemistry, it is a little different. You synthesize a compound and you go directly. Whereas in plant-based research, most of the journals will expect you to give a kind of how did you come to this conclusion that this possible molecule has certain type of activity like that? So it is better also to start from the low and move to upward.
Yes, sir. Thank you, Father. Thank you, Father.
Father, Dr. Johnsy Mary, Head Department of Chemistry from Auxilium, she has got a question. Ma'am, please.
Yeah, welcome, please.
No, yes, Father. I know that research with plants and plant products is very tedious and time consuming, and it needs a lot of patience and lot of knowledge also. They have to go through the literature and all that. But these days, what I find in students is all of a sudden they want results. So when they don't get results immediately, they give up. So I want, as you have got a very rich experience in research, so I want you to say a few words of motivation to youngsters as to how to go about research.
Thank you. Well, the only first sentence I would like to say is shortcuts cut life short. So we young, people may think, okay, we must get immediate results and so want to try something this way, that way. Well, all that will not work. Patience and hard work only ultimately will bring success in our life. So it may take some time to find the result, but ultimately when we are able to get it, that will be the greatest joy. Take, for example, Mr. Thomas Alva Edison, when he wanted to find a material which burns but does not get burnt. That is how we have got the bulb today. He had to try 1,000 and odd materials before he reached the last one, which was working for him. So the failure does not mean that we are really failing. It is only an indication that something is not working. So we will move in for the next one, and ultimately there will be always a solution. No doubt about it. God has kept some solution for everything somewhere. It's up to us to apply our mind and patiently work on, particularly, research is an area which takes longer time, long period and so on. And people have to understand it. I understand particularly students today, particularly lady students, maybe because of their life sketch and things like that, they may like to finish fast. But I think patience and hard work is very, very important. Thank you.
Thank you, Father.
Father, Dr. Xavier, Head Department of Chemistry, has got a question to ask. Yes,
welcome.
Sir.
Thank you, Father. Thank you for our wonderful lecture, Father. Just to add, Dr. Jansi Madam, I want to say something. In each and every local area, there are so many herbal plants are growing automatically during the rainy season. But in the modern agriculture, we are using herbicides to destroy them. Collectively. So there are so many herbal plants that they are coming up automatically with the blessing of the rain and the virgin of the soil. But in the name of agriculture, we are destroying our ancient capabilities. So we have to motivate our farmers in this field, and also there should be some marketing, some profitable line in the planting and farming the herbal plants.
Yeah.
So in this line, the government may encourage the farmers and also the local community. So people like you are in the higher position, you can also take this to the government for that.
Yeah, thank you. Thank you for the suggestion. You are very right, and certainly use of herbicide and so on should be discouraged. And as scientists, we can try to develop natural, plant-based herbicides, which will not unnecessarily harm the others. And things like that can certainly be worked out, and we will take it up with the higher bodies whenever possible to indicate to them that the agricultural sector has to be more careful with regard to the use of herbicide and things like that. Another biggest worry is the use of pesticide, for example, pesticide to control insects. And in that sense, I'm very happy we were able to come out with a natural pesticide, which is now prevalently used in particularly in Tamil Nadu and many other parts of places where they are exporting materials for outside the country. And it is purely herbal and very effective. So I wish many of our younger scientists can think in these lines and try to come out with natural materials, herbal-based materials. Here, for example, recently we came out with a herbal sanitizer, herbal hand sanitizer for corona. Suddenly our lab developed and we were very happy, and one company is now producing and selling it in a big way. And like that, if many herbal products can be brought out through research, then I'm sure it will contribute much for future generations. Thank you.
Thank you. Thank you. Thank you.
Further, great that you have been able to do this. Further, two or more questions are there. Will you be able to take up
please,
further?
Please, yeah. Please.
Further, repeatedly, two or three people have asked this. When a plant contains flavonoids, can you suggest some, methods to find out them whether the flavonoids are present or not?
Oh, very simple. There is a methodology by Harborn, preliminary identification of chemicals that are present. So it's simple method to check for the presence of various types of groups of compounds. And kindly follow that Harborn method, and you can easily identify whether the particular group of compound is present or not. Yeah. Thank you.
Thank you for that. There is another question from Balaji Janardhan asking for some web databases already available to survey the work that has already been done.
Yeah. Quite many are there. The Google search will give you any amount of material and information on the literature that is available. PubMed will give you, Scopus will give you. All these are, databases where already published literature is available. So kindly check any one of them and slowly get more knowledge on a particular topic that you are interested in, and ultimately you will be able to find some good progress. Thank you. God bless.
Father, one more question from YouTube, Father.
Yeah.
Sakti Priyadarshini asked, please tell us the mandatory structural elicitation characterization method of isolated compounds from extracts.
Yeah. How to characterize the compound that you have isolated. Very simple. Once you have a pure compound, check for the purity using any kind of methodology like HPLC will give you, the peak is very high and pure, it will give you 99 point some purity. Once that is there, you kindly take it for what is called a NMR analysis. If it is a crystal, you can directly go for XRD analysis, or the normal 2D and 3D and carbon and all that, NMR, CNMR and all that will give you the spectra which can be interpreted by the phytochemist who is well versed in identifying and making the structures elucidation with the help of the spectra that is available. So that is the step you have to take. Once you have a compound, check for its purity, then hand over for analysis related to NMR or things like that, or XRD, and ultimately it can also be, you can sometimes also give it for LCMS they call, that is also will give you some indication, but the phytochemist will suggest what are the things that are to be done in order to get to the exact and final structure. So it is always better to consult a phytochemist and then proceed for your work. Thank you.
Thank you, Father. Dr. Wilson Bosco Paul, he has got a question to ask you.
Yeah, welcome.
Father, good morning, Father.
Morning.
I have a small doubt regarding the plants consuming as raw materials. Sometimes some of the plants we have consumed, it will generate some internal heat to the body.
Yeah.
What if we isolate the compound, and will it create the same effect to the body?
The whole plant contains many molecules. So we are not very sure which of the molecule is bringing out that particular, and that is why we go for what is known as the bioassay test. So if that particular molecule is the one which is able to bring out the effect that we are seeing in our body, then naturally we can go in. That is how many people find what is called the medicine, medicine. For example, diabetes, metformin is a compound which we are using for medicine to treat diabetes. The original compound for getting this metformin was obtained from a plant. Plant was being used by people traditionally, and scientists, particularly industry, went in to check, and they found a particular molecule, and they checked it with the animal model, and they tweaked it a little bit, modified a little bit, and now we are taking it as one of the good medicines for diabetes. So certainly possibilities are there. The compound which is obtained may also have full-fledged effect as we are having from a plant. And another example is artemisinin. For example, artemisinin from the plant was, the juice was used for controlling. And later on, when artemisinin was developed and synthetically also modified, it is used as a good medicine also to control malaria parasite. So it is depending on how we are verifying and using it, we can go ahead. Thank you.
Okay, Father. Will it be safe to use some of the medicines that is
Safety has to be tested. Safety has to be tested. So there are methods described by international bodies to do LD50, LD90. So this will indicate whether toxicity is there present for a particular crude extract or particular compound and so on. So we have to do that toxicity test in order to identify whether this compound is safe. So only when there is that safety possibility, then we should go ahead. No doubt about it. So there are tests which are to be done. And any paper where we are showing some kind of activity like this, we'll also ask for the detailed study using animal model for the toxicity study. So that has to be carried out. Thank you.
Thank you, Father.
Thank you.
May I request the participants, if you have any more questions, you can raise hand or you can do it through chat. Please do interact with Father, who has got a vast experience in this field of phytochemistry.
Father, I need to ask one more.
Yeah, welcome.
Father, for isolation, we use usually column chromatography.
Yeah.
Is there any other method? Because column chromatography consumes more solvent.
Yeah. Yeah, there is a flash chromatography, which is more costly, where much less solvent is used, but isolation can be done also effectively. There are companies which sell these products, flash chromatography, very efficient in isolation and so on. You can kindly check on that, and if it is feasible, we can consider it. No problem.
Okay, sir.
Yeah.
Okay.
Father, I have a question on choosing the plant. When the same plant grows in different regions, does it make valid to claim that plant to work as a novel work?
In the sense, novel work in the sense?
No, when two plants in two different regions are being investigated, the finding of one plant, can it be claimed as a novelty if it has already been done in another part of the world or the country?
We can only say that particularly this work which you are doing is little different from the other one, then you can say it is a novelty. But by the plant growing in one place and this is the first time you are taking it from Tamil Nadu and you are analyzing, may not be accepted as a claim for novelty.
From your experience, how do you, I mean, what's your opinion on this further? Plant varieties, when they grow in different places, completely different temperature, things like that, do they have a difference in the phytochemical composition?
Yeah. That is there. That is surely there. Phytochemical in the sense when the quantity may vary. In certain areas, it will be little less. In certain other areas, there may be more. For example, we have done a big analysis on availability of azadirachtin from neem plants, the whole of India. So when we take a neem plant from Bihar, a neem plant from Mumbai, and a neem plant from Tamil Nadu, and within Tamil Nadu from Tuticorin or from Chennai, there is a lot of variation in the azadirachtin content. So that is certainly because of the soil, I know that is prevalent and so on. And that variation certainly will be there, no doubt about it.
So one question from YouTube, I think.
Yeah.
Most of researchers avoid aqueous solvent for phytochemical analysis. In that case, can we use for nanosynthesis?
Nano is one area of research. It is a little different. You know, the best thing is to go in for methanol if that is possible. And aqueous extract is ideal also because that is how we normally consume the traditional materials or plants when we are taking into our body. So if we are able to get aqueous extract and then find a working solution, that is much better. Otherwise, the methanol may be a suitable solvent. But nano work, I think it is a little different. Nano-oriented work is a little different. Yeah.
Father Sri Hemanta Upatyaya,
Yeah.
they have asked one question. For oils, after a plant oil is being extracted, how do we do the analysis? Is it the same like any other extract, or do we need to do some special test?
Yeah. For oil, we need to dissolve it in a particular solvent again. So check whether that oil is solvent. Where normally in different types of oils will be able to dissolve in twin 20 or twin 80 and things like that. So depending on how much we are using, we give that and then make use of it for analysis. No problem. We must choose a solvent to dissolve the oil. Emulsifier. I call it emulsifier, not a solvent. An emulsifier to dissolve the oil and then give the concentration of the emulsifier and then do the test. Oil naturally will not disperse to do the bioassay. So we need to dissolve it in an emulsifier and use a natural safe emulsifier and then do the bioassay.
Thank you so much, Father. Thank you. I'm
Thank
just
you.
waiting for any more questions. Are they being? Yes, Father. We have another one question. How to? Just a minute, Father. I am just rephrasing the sentence. How to collect a herbal plant in forest or agricultural land to find? They have typed a documentary. I don't. I think it has to be rephrased. I think they mean to ask whether we should get permission or something like that. If you are going to get it from.
If you are going into a forest department permission is needed. If you are selecting it from a common place, it is no problem. And today with the National Biodiversity Authority, which is established in the country, even if we are doing research with a particular plant and when we are going to file a patent or when we are transferring to another country, we have to get the NBA permission and so on. So there are a lot of rules are there available now. So kindly check for all these guidelines in the web search, which you will be able to get. But most important thing, for us, we cannot directly enter and collect. We need to get the department's permission to collect any material, whether it is plant or animal or insect, whatever, from the forest department.
I think they have, since they've used the word documentary, maybe for documentation purpose also we should get the permission for that.
Yeah. Even documentation. For example, we are preparing a flora and fauna, and every time we have to write for a letter to the director of forest department and get permission.
One final question maybe further. Someone has asked this. When plants are extracted by different solvents, do the phytochemicals present in it changes? Will it get modified due to the effect of the solvent? Aravindan has asked this
No,
question.
normally does not happen. But if you are allowing a column chromatography to be simply standing there for so many days, and then you do not do the work necessary to simply elute it as fast as possible, then there is certainly possibility for some kind of structural modification and so on. So if you are doing the normal type of work, it doesn't happen. And then after taking it also, you put it in the storage for particular temperature and things like that, then minimal changes are expected to occur. Normally it doesn't happen. As I said, if you are simply putting it and making it, leaving it like that in the column for 5 days, 8 days, or 10 days without eluting and things like that, it will affect.
Father, one final question from Mr. Napoleon.
Father,
Yeah.
for
Extraction,
that is one method suggested. Extracting the same product in different solvent, starting from NX and low polarity to high polarity.
High to low, yeah.
Will that be useful in isolation of plant? Will it be easy when you run the column? Is it a successful method?
Yeah. Normally it is done so. Start from the low polar, then go to high, so that it is able to bring out the different weighted molecules along. It is the normally used method.
Okay.
Okay.
Mario has raised hand. Mario, can you ask?
Just a minute.
Yeah, please.
Mario, please ask. Yes, Mario.
Father, good morning, Father. This is Mario, Father.
Good morning, welcome.
Father, thank you for the wonderful lecture, Father.
Thank you.
Just a very simple question, Father. It's just out of my curiosity.
Yeah.
Father, is it the goal of phytochemistry to extract compounds from nature and use them for medicine? Father, like it's the same thing that is happening in the case of the present trend of pharma. What they do is they take out a chemical compound and they give it for various ailments, Father.
Yeah.
So like I don't understand what is the difference, Father. Are we doing like is it that our incapability of synthesis of compounds is that we are going for extraction?
No, not that way. Not because we are not capable of synthesizing compounds. There has been a traditional use of a plant for long years, which has not shown any toxicity or any side effects, and therefore getting compounds from these to use for certain ailments is better than first in man-made synthesis of compounds and using it. That is the basic philosophy behind going in for isolation of compounds from herbal materials and so on. So it's not because we are not able to synthesize, it's only because there has been a tradition and has been in use for so long without any side effects and so on. And it is better to make use of them in our medical terminal than in going in for first in man-made use. That is the philosophy.
Okay, Father. Okay, Father.
Okay.
Thank you, Father. Thank you, Father. Thank you, Father.
Thank you. God bless. Yeah.
Father, it was so wonderful listening to you and the way you answer the questions. It makes us really interested in the field of photochemistry.
Thank you.
I believe the same would be the feeling of all the participants. So with much joy and gratefulness, we would like to thank you for your availability in spite of your busy schedule. To formally conclude, I invite Professor Angeline Mary to propose the vote of thanks. Madam, over to you.
Thank you, sir. Very good morning to all. I deem it a great honor to propose the vote of thanks to all who have helped us in making this section of the webinar such a resounding success. I would like to thank our distinguished speaker, Reverend Father Dr. S. Innasimuthu, SJ, for his thought-provoking and insightful lecture on scientific validation of some herbs used in ethnomedicine. Dear Father, it was a delight to listen to your deep insights in the field of phytochemistry, and we wholeheartedly thank you for addressing us in spite of your busy schedule. Thank you, Father.
Thank you.
Thank you, Father. I would like to place on record my sincere sincere thanks to Reverend Dr. R. Jaishanti, Principal, Auxilium College, Vellore, for her help in getting the resource persons and also for setting the tone for this webinar. Thank you, sister. I have my thanks to Reverend Dr. D. Maria Anthony Raj, SDB, Principal of our college, for his encouragement and support for this webinar. Thank you, Father. My sincere thanks goes to Dr. S. Jansi Mary, 8th Department of Chemistry, Auxilium College, Vellore, for her warm words of felicitation and for her great support for this webinar. Thank you, ma'am. I would like to place on my record my sincere thanks to Reverend Father Dr. Praveen Peter, Vice Principal, Controller of Examination, Sacred College, for his kind introduction about the resource person. Thank you, Father. I thank our respected and beloved HOD, Dr. Xavier Rajaratnam, for moderating the section and for the valuable guidance. Thank you, sir. I thank you all participants, and let us stay tuned for the next session to be handled by Dr. Jayakumar. Thank you, one and all.
Thank you. All the best. God bless. Yeah.
Thank you, Father. Thank you.
Father, once again, thank you
Yeah,
so much on
thank you.
behalf of
All
Sacred
the
Heart
best.
College and
Yeah.
Auxilium College.
God bless. Thank you.
Thank you, Father. So, dear participants, I hope you would have enjoyed Father's first session. Now we move on to the next one. May I now invite Dr. Scholastica Augustine, Assistant Professor, Department of Chemistry, Auxilium College, to take over the session. Madam, over to you.
Yeah, thank you, Dr. Collins. Can you hear me?
Yes, ma'am.
This is Dr. Scholastica. Welcome me one and all back to the second session of the second day's webinar titled Principles of Phytochemical Research. With immense pleasure, I welcome and introduce Dr. D. Jayakumar, Assistant Professor of Chemistry, St. Xavier's College, Palan, Kotai. I would like to place on record that Dr. Jayakumar was the first male student of Auxilium College and the first doctorate of the Department of Chemistry, Auxilium College. Dr. Jayakumar has published several research papers in reputed national and international journals. He has also published a book titled Lab Manual of Organic and Phytochemistry for Theory and Practice. His area of research includes isolation and biological studies of photoconstituents and synthesis of chemical compounds. He has done both his UG and PG programs in Lala College, Chennai. I wholeheartedly welcome you, Dr. Jayakumar, to this webinar series spanning for 3 days to deliver your talk on isolation, synthesis, and structural modification, schemes of coumarin hybrids as anti-cancer agents. I also take this opportunity to welcome the organizers and the learned participants for the session. It's now over to you, Jayakumar.
Good morning, madam. Thank you very much. Can everybody hear me, ma'am?
Yeah, sure. Yes,
Yes, ma'am. Yes, ma'am. I'll share the slides, ma'am. I hope everybody can see the slides.
Yes, sir. Yes, sir.
Okay.
Yes, sir. Full screen?
Yeah.
Yes, sir. Thank you. Sir, you can continue. Yes.
Yes, sir. Thank you, sir. Dear dignitaries, Secretary Principal, Head of the Department of Sacred Heart College and Auxilium College, I'm Dr. Jayakumar, Assistant Professor of Chemistry, Department of Chemistry and Xavier's College, Palan, Kotai. I express my sincere gratitude to the management of both the colleges for having this webinar, and I'm sure this webinar would inspire many research scholars and student fraternity. I extend this opportunity to make a special thanks to my mentor, Reverend Dr. Jaishanti and Dr. Collins, for having me here, and a special thanks to my Scholastica madam, and she forgot to tell that I'm her student too, Madam Scholastica. Having said that, we'll continue into the topic. I hope you can see the screens, therefore I'll continue. The topic centers around coumarin hybrid. So what is this coumarin hybrid? Therefore, to know about coumarin hybrid, first we'll pay some attention towards molecular hybridization, its scars and emergence and recent trends that are prevailing in this globe. We'll spend some time over discussing these things, then we'll jump into the coumarin. why we chose coumarin to make this hybrid molecule. Why not other natural scaffold for that matter? Because we have several hundreds and thousands of molecules, especially secondary metabolites. Name a few. Why not anthraquinone or tannin, flavonoids, even alkaloids? All these things are very famous for its biological activity. So what's so special about coumarin? To explore this thing, we now say something regarding coumarin. Then after completing into coumarin, we'll jump into the other section. We'll talk about the structural modification. The very purpose of introduction to structural modification is this. What are the ways and means by which we can construct or architect a hybrid molecule? And what are the tips and what are the nuances that has to be getting explored? Okay, then after we'll just make a brief discussion over it. Then we make 2 case studies pertaining to the said topic. And finally, as a bonus one, are we capable enough to construct a hybrid, not only coumarin, for any natural scaffold for that matter, in our own laboratory? Putting straight, this I'll show, or otherwise we shall discuss certain things such that we make an environment where we can do our own hybrid in our own laboratory with low cost. Okay, and I hope this presentation will be really helpful for the medicinal chemist or chemical biologist or even any budding researcher who wish to pursue their PhD work in this department. Thank you very much, and we'll go into the first slide. First, we're going into the World Health Organization's report. It says that globally, 1 in 5 male and 1 in 6 male are happen to be a victim of incidents of cancer at least once in their lifetime. And as far as mortality rate is concerned, it is 1 in every 8 male. We're talking about in a global scale, and 1 in every 11 female losing their life for this devastating disease. Okay, an alarming report says that in 2018 alone, there are about 18.1 new cancer patients being reported, of which nearly 9.5 million people lost their life. So this would really gives the devastating story of serious cancers. Somebody got a chat? I'll go into the chat. Okay, I hope you can, everybody hearing me. Therefore, I'm closing the chat and I'm continuing the topic. Okay, so there is a pressing need. Thus, there should be anti-cancer agent should be really created or designed such that it has its crucial for the treatment of cancer. Or otherwise, it is now imperative to develop a novel anti-cancer agent. So what do you mean by the term novel? The term novel should be centered around of these 4 parameters. To decomplex the term novel, it should have a high specificity. What do you mean by high specificity? Suppose we have a claimed or a drug lead molecule which would have an anti-cancer activity. Suppose we have a protein, this anti-cancer activity should be able to be binded so well, so strongly with appropriate attractive forces. Only then it can act as an anti-cancer agent. And moving to the other one, it should be a better or a suitable alternate to our conventional, that is surgical procedures. As far as many cancers are concerned, the prevailing treatment, or otherwise one of the very famous treatment, is a combination of chemotherapy, radiotherapy, and surgical procedure. But it has its own limitations. As far as limitations are concerned, this treatment is, is limited by the factor of healthiness of an individual whom we try to cure the cancer, and it also depends on the gender, maybe a male or a female, that also is also of question, and followed by age is also a question, and also finally the stage that we are really discussing. As far as 3rd stage and 4th stage, the success of this surgical procedure is somewhat of question. And therefore, if at all it's an anti-cancer agent, it should be a suitable alternate. Having said that, not limited by these 2 factors, there are some cancers such will have a problem of resistance towards the drug. Or otherwise, there is also another problem which I have not mentioned over here. That is, once if the cancer has been cured from a particular person, doesn't mean that he will not contract the same cancer anytime further. There is always reoccurrence possible. And therefore, we need to take into account that how do we tackle this multi-drug resistance cancer. And finally, all these things has to be discussed in a high mortality scenario. So this is what the pressing demands before us. Now, so we find here lots of scope and emergence and need for us to work in this particular area. Okay. So how does this drug has been really been designed so far? As far as conventional treatment is concerned, our existing idea is this: we take or we envision one particular molecule. This one particular molecule will address or act on one particular disease such a way that it will take one particular pathway. Or we simply speaking, it will be a single or solo targeting agents will be used to this approach was used in addressing the disease. But most of the biological disorders, especially cancer, do not happening by a one single targeting agent. It is more complex. And there is an interplay of many proteins. It's like a machinery. So many parts will move on its fashion in a coherent manner by which regulation of regulation of cell process takes place. If there is any defection in it, and the anti-cancer agent sure has to address this issue, should be in a manner that it is not a single targeting agent, but multiple targeting agents or the multiple pathway you should follow simultaneously. This is a very mammoth task. But this is where we researchers now making a paradigm shift from one molecule to addressing one disease through one target approach. From this idea, we now moving away such that the new idea would be one molecule or aggregate of so many molecules as one cluster, and that will address one particular disease by taking into multiple pathway approach, or otherwise multiple target approach. This is the idea that we are now we are facing in these current situations. So whenever we think about multiple pathways, we are now in the realm of molecular hybridization. So what is molecular hybridization? Making it very simple, it's nothing but a combination of two or more pharmacology into a single molecular architecture. So we just combine or conjugate these two pharmacophores. What do you mean by pharmacophores? It is a particular claimed drug candidate or a candidate or a lead molecule which has its therapeutic utility. So when we combine two or more molecules which has a therapeutic utility, definitely will be ending up with a single molecular architecture. So what should be the prime goal in creating the primary single molecular architecture? It should be kept in your view that we need to optimize the individual potential of the pharmacophores that are being binded. How do we really optimize? In what sense? In the sense that we increase the potential, especially we improve the affinity, which means the pharmacophore should be able to bind with the protein. That's the first thing. And it has to do its job of curing any therapeutic activity that will be beneficial for the patient. That is what the first paramount goal to be envisioned when you think about molecular architecture or simply molecular hybrid. So from now on, rather than saying it is a molecular architecture, we simply regard it as a molecular hybrid just because of the fact that we're binding to a more pharmacophores. I got a chat, just a moment. Degrading, dragging because of some network strength. If it's, I think you can hear me. Can you all hear me?
Yes, sir, we can hear. We can hear.
Yeah.
Yes, we can hear. Only now and then it breaks your audio. Your audio is breaking now and then. But otherwise, it is clear.
Okay. Okay. I'll continue. Next. So put it in a very simple terms of equations, we have a pharmacophore 1, which has some therapeutic activity or utility. It has been combined with another pharmacophore, which will be complementary to the pharmacophore 1, or it may be having the other sets of therapeutic beneficial activity. But rather, when they are conjugated, will be end up with a hybrid molecule. In other words, conventionally, it is a hybrid molecular architecture. So how do we really conjugate each other? There are many ways. Chemically, they can be binded, or otherwise, if not, it can be binded by using a linker, a suitable linker. This suitable linker is also called as a tethering agent. So tethering agent is one which will bridge two or more pharmacophores, and in addition of being bridging the two pharmacophore, it serves another purpose, that it also contribute for the therapeutic activity. So this is an added strength. So combining all the therapeutic activity together will end up with a hybrid molecule. Now, by giving the hypothetical idea, sometimes it may make us to believe or make us to skeptic that we make a sort of a tall climb by this kind of hypothetical question. Therefore, to validate, if it be a tall climb, we have a compound called Werloxin. This is the structure of Werloxin, in which it is really a hybrid motif. This hybrid motif is very successful in treating cancer, or perhaps it is at least passed some clinical trials for and becomes a very promising agent or lead molecule in the treatment of cancer. So now, as of now, we'll satisfy ourselves that this is, though being a tall climb, it is really workable thing. And how do we materialize this idea, hypothetical idea? We have to envision, and then we have to design, and we have to synthesize these hybrids. And we can do that by molecular hybridization technique or click chemistry methods. Now, having said that, why do we turn the attention towards coumarin hybrids? Lots of natural scaffolds are available, and we pretty well know that all the sessions we learned that natural scaffolds are very much good in binding with our body. Therefore, there is no surprising fact that it will not have any sort of adverse effects in our body. Number one. Number two, and we also seen in these presentations, or before presentation, father's presentations, or previous delegates presentations, that naturally occurring materials being used for a century of years, it requires no any preclinical trial. Rather, it is directly be consumed by human beings, and based on that trial and error basis, we label certain plants as a medicinal plants. Therefore, it will elicit. Therefore, there's no wonder that it elicits in biological activity. But still, why do we focus attention towards coumarin? Because there is a beautiful things behind this coumarin. Okay, so natural occurring scaffolds are being conglomerated with anti-cancer pharmacophores. Okay, so we have one pharmacophore which is expected to be, or otherwise declared, or it possesses already well-established anti-cancer activity. Now it is being conjugated with a naturally occurring motif, with or without a sort of a tethering agent, in a view to develop a multifunctional molecule, and one of the functional should be a cancer as of now, such that it has to be go with the reduced side effects, it has to overcome drug resistance, improved pharmacokinetics, and of course oral bioavailability. Because in modern times, just a moment, I shall stop your video so that your audio is clear. Yes, sir. Yes, sir. Okay. So as far as this molecule is concerned, we do have. Can you all hear me?
Sir, we can. Yes, sir, we can hear. Continue, sir.
Yeah. Yeah. So being very available natural scaffolds, we have chosen this coumarin containing hybrids because it really occupies an important position in the development of novel anti-cancer agents. And what is that important position? There are two questions that may be striking in our mind: whether are this coumarin is really a versatile compound? If so, why? And how it attains that versatility? These are the two questions that are striking in the mind. By answering these questions, we'll be in a position to choose one of the pharmacophore, a better one, so that it can be added with anti-cancer agent that will end up with a fantastic molecular architecture. Therefore, as of now, we'll now going into this coumarin. First of all, coumarin is ubiquitous in nature. It presents everywhere. Most of the plant kingdom, especially Rutaceae, Umbrophilo, and even Morinda species, are enriched with this coumarin. And also, it presents in a large quantity. Of course, there are various secondary metabolites which even spread in the plant kingdom, yet the coumarin is the one which presents in the large excess. And also, in my personal opinion, I would say that it is somewhat relatively easy for us to isolate and characterize. And therefore, it present in the large quantity is being another added advantage in taking coumarin as one of the pharmacophore in making a hybrid molecule. And in this area, it also has another important area, that is its biological activity. Realistically speaking, the coumarin has a very big or very diverse applications, an array of activities ranging from anti-cancer and in with anti-inflammatory, so on and so forth. And apart from that, it also has low toxicity. So together, coupling, that is on one side having higher biological activity, on the other side is a low toxicity, it would be a better and beautiful choice for a researcher to try it with coumarin. And in addition to that, this is for our chemistry part. And these coumarin compounds can be readily prepared in a laboratory with less efforts. So taking all together, this coumarin would be, it's not a overstatement saying that it is a very privileged motive to study in the coumarin. Next slide. So now the question comes, is it so versatile? How can justify this particular point? See, the biological activities have been scattered between antibacterial, anticoagulant, HIV, antioxidant, antitubular, so on and so forth. This is a very growing list. It is not limited with the mentioned, activity here. The length still goes on. And of course, the therapeutic property completely depends on the nature and the basic substituent present in the molecule. So I think it is the right time to number them. This is how we will be numbering. This is what somewhat contradictory, where we will be using the numbering only for this carbon skeleton. And as far as the heteromolecule coming into the ring itself, it's better to start numbering from here. So there are certain regions, for example, this region, or I would say that this region and this region, actually every corners has a very interesting properties. We'll be looking into why they are showing such interesting properties, but these are some of the easy regions. What do you mean by easy regions? For attaching any group along this path, or here is somewhat much easier. Okay, and all these things making this molecule so versatile. Next. Why it is so versatile? The answer would be somewhat very, very simple. So I will give a couple of seconds. Somebody can guess and tell me what makes this so versatile, or how it becomes so versatile. Rather, we have various compounds, because I would say that my personal compound would be anthraquinones, because I've been first tried making hybrids, making use of anthraquinones. But still, I found that coumarin would be somewhat better than that of anthraquinones. And going into the reasons behind that versatility, it really, made me ashamed because of having simple reasons. So I'll give you a couple of seconds for research students, if there is anybody who could come up with the reasons, make some wild guesses. Okay. If there is no reasons, I'll continue. Okay, I'll continue myself. This is because of smaller interactions, molecular non-covalent interactions like pi-pi interactions or hydrophobic interactions, or sometimes even electrostatic interaction. These are two contrasting these are two contrasting effects, but still these exactly present in every or at least almost all coumarins. And these two interactions are the main cause. I think it's my hunch, but doesn't mean that this is a very correct answer, but I make my hunch that these are the two interactions that really responsible for most of its biological activity. Apart from that, hydrogen bonding is also one of the well-established reason for the biological activity. Of course, metal coordinations. These two things have been, even I personally experienced the influence of these effects in my own research work. I'll try to show at the end of my research. So what we want to mention over here is this. These weak van der Waals forces makes the molecule coumarin to be easily get binded with the protein or any cells of our interest. It has very easy to catch on to be get binded itself, and it's starting doing its work. What is its work? It's eliciting some biological activity that will be beneficial. Next. So we've been now moving into the various schemes of by which we can make an hybrid. To address that thing, we pretty well first know how this anti pathways are been moving along, and how this the expected anti-cancer agent should strike it off at the right end, at the right spot, such that there is no any cancer proliferations. There are various pathways. For example, I would say that there is a pathway. Yeah, just a moment. Give me a minute. We have a pathway 1, pathway 2, and 3. These are the interesting pathway wherein which anybody can come across in every published journals. Other than that, we also see in recent times there is lots of research taking place in the shock proteins. Okay. So all these the other things are enzymatic pathway, very complex, but still there are lots of research going on this field. Fine. So how this coumarin actually working is, there are lots of proteins and lots of enzymes that are doing their regulatory work such that the cells grows. Now, protein kinases like AKT, MAP, these are the kinases which undergoes for the cells regulatory process and growth process. And that is cell, it will helps the cell to get divides. Now, when there is any dysregulation happening here, eventually they will, rather than going into regulatory pathway, they will turn into a cell proliferation. Therefore, that will leading into the next stage of cancer. Now, our coumarin either stops here, rather than going into a cell proliferation A stage, it will repair such that it will go into the normal stage, or otherwise it simply stops or prevent the cell proliferations. So how it prevents cell proliferation? Actually, these are two different parts to my understanding, but still I don't know why they have been given a single entity, but this apoptosis pathway is one and cycle arrest is maybe the other thing. Whereas the apoptosis is otherwise called as programmed cell death, which means the upon administration of your anti-cancer drug or coumarin as of here, they will, rather than going into a division process, they make the cell or insist the cell to commit suicide. That is called programmed cell death. And once this has been said, the cell will no longer survive, it will dead. And thereby, there won't be any proliferation of cells. This is one way of pathway. Rather than that of making the cell to go die, it will just stop the cell division. In a cell cycle, we can put it in a form of a cell cycle. The cell starts its life here, and it will grow and will do its work. And after the stage, it has choices whether to take a apoptosis pathway or will it take a division process. So, division process. There are been life cycle can be drawn of a cell. Rather, there is a midpoint making the decision, crucial decision, whether to be get divide or apoptotic pathway. We call it as a checkpoint, GS2 checkpoint. Yes, but G checkpoint. In that checkpoint, if it was cell, any anti-cancer agent make a climb to stop there, then the cell will not divide at all. It stops there itself, whereby we can arrest the cell growth. This is two pathways. Apart from that, this is the one which we'll be seeing in our in a case study, where in which these alpha tubulins are in a regulatory process which needs to be get polymerized, which will further make the spindle fibers to go across, which is helpful for the cell division. Here, the coumarin will prevent this polymerization. Once this polymerization is being prevented, they no longer can, the spindle fibers can move at the opposite poles, which paving the way for cell proliferation. Thereby, also you can stop this process. Likewise, it can go and repair the DNA which has been affected or get modified, or it make the DNA to decay. Thereby, the cancers which are being connected with the DNA can also be arrested or stopped. And finally, there is a protein called shock protein, and this protein will change the normal cell into a invader, cell invader, and this will further creates the problem in the cell normal process. This coumarin will prevent this shock proteins. So this is how we find the various pathways by which the coumarins can do its job. Now, these are some of the motors. Say, for example, we have a coumarin. It has been binded with sort of butene type of moiety and resulting a suitable hybrid. Likewise, we find here coumarin binding with histamine, icetin with a tethering agent. What do you mean by tethering agent? This is tethering agent. This will really bridging two, that is coumarin and your icetin molecules. Therefore, by making these kinds of hybrid molecules, and there are various tethering molecules and other pharmacophores available, everything has its own significance, and everything has one sole purpose to address the anti-cancer activity, of which we'll now take only this study, only this case, how it really stops the cancer. We are not going into details of how this molecule really just a minute, how these molecules really been prepared. Rather, we are interested in how efficient is it in its job. So Singh and et al. Synthesized a series of novel icetin coumarin hybrids using triazole as their tethering agent. Why they have chosen icetin? Because icetin is a very famous pharmacophore for anti-cancer activity. So what happens when a pharmacophore, which has been already recognized for anti-cancer activity, being binded with a natural motif? It will test it. A series of compounds have been prepared, and it was tested against a panel of cells. So in this panel of cells, it was found that the results revealed that there are three compounds. Let it be named as 1, 2, and 3. The structures will be shown shortly. The compounds 1, 2, 3 were found to be very effective against all the cells in this given panel, except the prostate cancer, that is PC3. Okay, this is the code for the cell line. And therefore, it was found that icetin moiety with a thiolinker is also one of the contributing factor for its anti-cancer activity. This is also an added information that can be revealed out of this particular study. And now, so let's see it in a form of a graphical thing. We have icetin molecule, coumarin. They have been binded via triazole linker or a tethering agent, such that we end up with this compound, end up in a series of compounds. And how these compounds are vary? They are vary by the length of the tethering agent. You would have been seeing here by the length of the tethering agent. There are 2 carbons, but here we find 3 carbons. So, and also by the substituent being attached to some positions, even in the pharmacophore itself. Now, and these 3 compounds seems to be promising results, especially against the cancer THP. What is that cancer? Just a moment, previous. That is leukemia cancer cell lines. So it was very active against leukemia cancer cells. And especially this compound 1 is shown the best result of the value IC50, 0.73 micromoles, whereas 2 and 7 having little value, larger value than these things. Okay, now what is the rationale that can be given for its huge activity against various cancer cell line? That is because of molecular docking studies have been really studied. I'm just giving the glimpse about the results alone. Rather, as far as molecular docking is concerned, I wish to present my own results. Therefore, I'm just giving sort of a glimpse over here. You would have seen there is a pi-pi stocking attractions here and polar attraction here and there, and hydrophobic interaction in the other sides. Though this helps some way or the other way able to fit into the host or a suitable protein of our interest, and that by it starts eliciting its behavior. So it's very easy to be get binded, and because of its low toxicity, it shows its work. That is how it wins the race. Now, as far as chemistry is concerned, we rather not caring much about the result. Rather, we really interested in how do we achieve that result. So we delay on the mechanistic pathway. Going into the mechanistic pathway of this molecule, it was found that they are gone into a tubulin inhibition. Short before, we studied about various pathways, of which one is the tubulin polymerization, and this particular molecule somehow stops this tubulin inhibition polymerization. That is tubulin polymerization is being inhibited this particular molecule. Okay, and that was confirmed with the Western blot method. Amongst that, it was the compound 1, which shown a very better results of about 1.06 moles, micromoles, sorry, and therefore this compounds, given rationally about their anti-cancer activity. And these results combining together says that it was a very suitable molecule for an anti-cancer agent by having inhibited the tubulin formation. Now, going into the other case study, we have fluorescent hybrids. So the fluorescent hybrids is really a source of nitrogen oxide. And this excess of nitrogen oxide is expected to show anti-cancer agents. It's a known fact. And therefore, when you take this fluoroxene to be an anti-cancer pharmacophore, what happens when it is being binded with coumarin or conjugated with coumarin? Of course, it was studied in two different scales. One is in vitro scales, the other is in vivo scales. In vitro scales, it was studied by using MTSA, and there are a series of compounds being formed. The results revealed that a particular molecule to be labeled as 52 exhibited highest activity. And that even the drug-sensitive HeLa cells, and of course, the other cells are also being tried, and it shows a better results. And the mechanistic pathway studies reveals that it now elicits cytotoxicity by stopping or by inducing the apoptosis or by stopping the cell cycle. That is G2 by M phase, especially in this cell line. And therefore, it also disrupt the MKA1. These are kinases which will essential for cell regulatory process. So by correcting these things or stopping the phosphorylation, by disturbing these things, the cell was not proliferated any further, thereby it act as an anti-cancer agent. But still, by just having the better results with in vitro study, we cannot argue about the same will reproduced in an in vivo study. So if you want to talk about anything have anti-cancer activity, it should be on one hand should be a good performance, shown good performance in vitro scale. On the other hand, it should also shown the better results in the in vivo scales. Being time hurrying up, I'm rushed through. Therefore, it was tried with the in vivo rat model, taking about 15 milligram per kilogram, and the compound 52 has shown some tumor growth. I mean, it inhibited the tumor growth, and that inhibition rate is about 55.8, exactly doubling the concentration for another group of rats. It was proven that the tumor growth was further suppressed, and the inhibition rate was increased to about 65.5, probably around some 0.25 times higher than that of the previous scenario. So also it was observed that added information that there is no loss of weight in the mice, and which means it has not less toxic, otherwise no toxic at all, which was further given with the corroborative evidence of lethal dose treatment, which stood as 263.6 milligram, which means it has a very good anti-cancer activity and low toxicity, and therefore we can definitely, or we can say it is a promising compound or a drug lead molecule or a better candidate, whatever we say in that words. We say coumarin fluoroxene hybrids is workable thing. So this is the structure of 52. For some reasons, I also drawn 53. That is because I want to show about the structural activity also working here. But now I find that because of time lagging, I'm just skipping these areas. Now we're moving into the final session. Can we construct such a hybrid in our own lab? Can we do that? Of course, we can do that. How? I'll give some tips. That would be a sort of a tips. Number one, I'm just taking my own example. I don't, I mean, I haven't given my current work for a reason that was yet to be get published because of this COVID pandemic. There is extreme delay. Therefore, I've taken some of the extract of my PhD work and I show it to you. With enlightened, I mean, my guide's idea, I have happened to choose Miranda Pubescens. I dedicate my sincere thanks to her because of her only I was introduced to this particular miracle plant. Okay, this miracle plant is extracted for various secondary metabolites using different solvents. There are various methods available, of which I just go on into percolation. What are the other methods available? We have out percolation, we have countercurrent ion, and we have liquid partition. It all depends. But this cold percolation is somewhat cheaper, and of course, it is a time consuming thing. But still, if we, for any beginners, this would be the easiest way by which you can really learn lots of skills. And therefore, what I want to say here is you take a plant, you crush it down, dry it up, crush it down, and take a big, I mean, large vessels of which you just pour the solvents and you separate that liquid part. We call it as a macella. Then once again, the same dried extract will be refilled with the chloroform successfully with ethyl acetate and methanolic acetate, so on and so forth, which will be adding various extracts. And upon taking the extracts, I mean, it has to be dried to 1/3 of its volume, and it can be now purified or separated by using various techniques I used with column chromatographic technique. As of now, we have a very recent technique. Say, for example, we have a flash chromatography, which is even available in our own college. St. Xavier's College Chemistry Department do have a flash chromatography. And other than that, we have HPLC. We have a HPLC or a GC being hyphenated with mass spectrum, such that you will be injecting on sample in one end and you will be happen to instantly purifying or separating the compounds, and you can instantly identify the compound. That becomes so easy job in these days, but still it was expensive and it is its own limitations. You cannot inject any sample just like that. Therefore, though this column chromatography being a very primitive one, this would be somewhat skillful. You can learn informations and it was very cost effective. The only thing is it will time consuming, of which I have taken the XLS extract and I've just purified 18 fractions, of which there are 2-3 compounds I separated in a purest form, and that interestingly would be coumarins. So these are some of the data were used to characterize my 4-hydroxycoumarin. I'm rushing on here, and it is a proton and a C13 NMR for the isolated coumarin. Now, this is how I isolated natural coumarin, but still I was inspired by this coumarin. Actually, as of my research is concerned, I was more drawn towards quinones, but then while I'm studying about the ways and means of isolating coumarin, I learned lots of interesting information that really inspired me to make these compounds, and therefore I tried to make these compounds in the lab itself. Sometimes or otherwise you have to isolate the compound, you have to partially synthesize it. As far as coumarin is concerned, you can see the molecule and there are lots of methods available such that you can take this molecule, or otherwise you can make biomimic the same molecule what you have been seen in the plants. Various methods available. Say, for example, Peckman's method or Novo-Nigel's methods are the very common and interesting easy methods are available to make compounds. So these are the research articles which I cling to. I just simply copied the way they give it. Definitely will be ending with the same coumarin if you follow these methods. Okay, by relying to this method, I prepared a series of coumarins over here, and of course these are its characterization part. And of course, if you want me to stop it, I'll stop it. If you wanted me to stop it, I'll raise your hand. Otherwise, I'll go into its application part. You can raise hands if you wish.
You can continue, sir. I think, can you take 10 more minutes, I think. Yeah? Can take, sir.
Yeah, because I have plenty to go on. If I have time, I'll come back and characterize this one.
Yes, sir, you can continue.
Yeah, it's okay. So going into this 8-nitro-4-methyl hydroxycoumarin, it has been, of course, I synthesized in this lab, and we gone for GCMS. All these things is taken in the Chennai, IIT Madras, and something to my friends from foreign, and I have got the data. So the peaks at 221, and on the other data are helped us to calculate its molecular structure, which centers around C10H7NO5. Of course, these things are highly relied on library values, because for any established compounds, you would have having series of speaks that you can look for the library match with the database. There is a database called SDBI. I write it here. There is a database called caps lock SDBI. This is a Japanese database. There are lots of database available as far as somebody who've been working in molecular docking or the work which was much related towards biology. You have a protein database, and there are many databases available, and you can access this database. It is actually free of cost. When you're going into accessing this database, definitely that would be really helpful for you to. Somebody's been making mark. What do you want me to do? I think you may be finding difficult to read this thing, because I made a mistake when I doing my PhD. I didn't get that original file, so I get the only the PDF version or get the printout. So that's the problem behind it. I'm sorry for this weak report. Okay, my point over here is this SDBI will throw lots of informations, and the library match will help you to resolve your compound. And these are my proton NMR and C13 NMR, and unambiguously considering the literature surveys, we been found that this molecule would have this type. And also, this is the molecule we intended to prepare, and therefore there is no wonder we have been ended up with this particular molecules. So this is the data for a given molecule. Okay, now we'll entering into the very important area. So far we have an isolated compounds, and based on the inspiration, we have synthesized other set of compounds. Now, how to say or assess its pharmacological activity? That's very crucially important, because if you are good enough in characterizing or labeling it to be a promising compound, then you can go further. Or otherwise, it would be a mere waste of time, or otherwise you won't be successful as expected. And therefore, you should have a very clean approach in systemizing. There should be a very proper systemizing approach to assess the pharmacobehavior or biological activity of a given compound, of which, based on the literature survey and some wise men's advice, I devised this method. Of course, my guide also helped me, taught me to how to set this information. Thanks to her one more time. I also thank Dr. Augustine, because he is also a person who helped me in making all these preparations. Now I'll continue here. First, I set a antioxidant benchmark. I synthesized a series of compounds or isolated. Just a moment, I got a chat. Participants can post your questions in the, okay, fine. I'll continue. As far as a series of compounds being isolated or being synthesized, the first thing you have to do is, if you want to look for its anti-cancer activity, you better go for its antioxidant potential. Doesn't mean that any anti-cancer activity should essentially have an antioxidant activity. There is no any strong correlation between these two, but this is a sort of a benchmark which will allow or filter out some untoward molecules. Of course, it's a guideline. It is up to you to judicially decide whether what type of antioxidant you wanted, or whether you skipped one and going for some other things. But as far as my research is concerned, even today, I stick to this strategy yet. That is, I take a simple antioxidant test, and I just screened all the synthesized compound, all the isolated compound for its antioxidant activity. But rather right now, I would say that rather than sticking to one particular activity, it is better to go for a panel having at least more than 3 tests. Say, for example, right now I'm trying with FRAP method or ORAC method, and ORAC method, and so on and so forth. But I'm just a beginner in this method. I don't think how far I've been succeeded in getting the results out of it. But of course, I have quite a little bit exposed to this TPPH method, and of course, this is also one of the interesting method to as a benchmark for antioxidant activity. Therefore, the take-home point would be, rather than sticking to one particular antioxidant assay, it would be a panel containing 3 or 4 would be better if you really could afford for it. Once, when you have a series of compounds which qualifies these antioxidant stage assays, then which means qualify means it becomes some significant results. Then from then on we call it as a candidates, and these candidates are tested for cytotoxicity, because cytotoxicity is one of the crucial factor to determining the anti-cancer activity. every compound should have anti-cancer activity only by via showing cytotoxic activity, because after all, the most thing is it has to kill the cancerous cell. Having said that, as I told you, you can have many assays for that method. For example, tryptophan method or any other method are really available, dye method. But in those times, because of some personal reasons, other things, I just stick on to only one particular assay, that is MTT assay, methyl tetrasodium salt assay. Then and the candidates which shown the better results of cytotoxicity is now intended to study how it gone into cytotoxicity, because cytotoxicity is sort of a screening test. By having the results of cytotoxicity alone, we cannot argue, or our argument will not stand for its real anti-cancer activity, and therefore we need to show the evidence, or we should convince ourselves it has killed the cell by a known mechanism. There are lots of mechanistic pathway. For example, in my research work, I cared about the caspase protein. What is this caspase? It is a cysteine aspartate proteins. Does the property of tethering agent affect the coumarin hybrid compound? Of course, of course. In one of my case study, I would have told you that compound 1 and 2 and 3, I'll show you at the end of the thing, where in which the length of the tethering group also contributing. In one time, the tethering agent will be smaller, and the other time it will be in longer. Therefore, the length of the tethering group and the type of the tethering group and where it is being binded, everything is to be taken considered, to be get considered when you talk about anti-cancer activity. Okay, now continuing over here, this is cysteine aspartate proteins. Okay, other than that, we have a BCL practice, BCL proteins, or we have the other one. Lots of studies are available. Brax protein, and the other things, of which I just going into the caspase. Caspase is not a single protein. It is like a cascades of a series of proteins or enzymes packed together. Say, for example, we have caspase 3, caspase 9, caspase 2, caspase 12, 14, so on and so forth, of which we just focused how this caspase 3 and caspase 9 being reacted upon administration of our drug lead molecule, which becomes a better candidate out of this antioxidant activity, because this caspase 3 and caspase 9 is concerns with the apoptotic pathway direction. Therefore, we study the caspase proteins and followed by gene expression, because this would be a corroborative evidence for to support your claim. As for gene expression studies, I relied on P53. In recent times, there are various other methods available, for example, flow cytometry, and the other methods are also very readily available. You can even go into that method. That was more sophisticated method and more reliable method too. Okay, and as far as my research is concerned, the workflow goes on. I take the series of compounds I tried with antioxidant activity, which shown a promising results, were taken into considered as a candidates, taken into cytotoxic activity. The one showing the better results are being now studied for the mechanistic pathway, say for example, caspase proteins and gene expressions. But I'm not limited over here. Okay, so these are the ways by which I did my antioxidant activity. These are the results, and the results have been considered in two parameters. At the maximum concentration, for example, 800 mic. This was too much for a result to go on, but still I tried because I want to know where this plateau been reaching. Therefore, I tried up to 800 maximum concentration, whereas my primary results relies on these IC50 values, and it says that 7-hydroxy-4-methylcoumarin seems to be the better molecules. And of course, it's not only the antioxidant results alone have been taken into consideration for cytotoxicity, various other parameters too. And as far as cytotoxicity, this is the protocol I adopted, and this is my results. So I just compared the results of cytotoxicity of both, which one isolated compound, that is 7-hydroxycoumarin. Umbelliferin is the name of the common name of the particular coumarin, or otherwise I just synthesized a similar type of coumarin and I compared the result. The synthetic one turned out to be better, showing better result than the natural isolate one. Then again, I going into the caspase test and finally into the expression. So the caspase proteins says that the untreated cell, when compared with untreated cell, which means the cells which is not been administered with my drug or a drug lead molecule at least, and with increased at the maximum concentration, the caspase levels was expressed to about comparing from the normal level, it is about 1.5 times been increased. Just a moment, I got a chat. by which mode we will get the, okay, fine. So by doing this is about 1.5 mole and 1.5 to 4 moles, we found that the coumarin shows a greater activity. Now, RT-PCR results indicates that this is the lane by which the P53 gene being found to expression was studied by the admiration of a drug molecule, which shows that there is an increased expression, which means it really induces the apoptic activity. And recently, I'm also subjected the same substance or much relevant substance for a molecular docking study. And the enzymes that I was taken into consideration is monoxime oxidase, which will be really helpful in making the serotonin and nephrotin. These are related towards the Alzheimer's diseases. Alzheimer's disease is also sort of related with the various function of proteins, and therefore I tried for this how these things are turn out to be. And it is also known that the coumarins play a key role in making Alzheimer's disease too. Therefore, I think it is not out of place to study Alzheimer's disease or the proteins that are concerned with Alzheimer's disease in terms of coumarin. And you would have seen there is a picture which depicts sort of yellow dots over here. And this yellow dots implies, of course, the drug molecule has a greater ability to get binded with the target molecule, that is the proteins, and how it is being binded. This would be a sort of a simple or a mere hydrogen bonding. Okay, so this is how the molecule is able to bind with the protein molecules. It has been seen in the different scales, different regions. Autodock method has been used. Now, so the current investigation determines that there is a probable binding take place between the sample and the proteins, whereas HMC stands for hydroxymethylcoumarin. HC stands for hydroxycoumarin. This is a natural one. This is a synthetic one, and it was found that these monoxidase have been, there is a change in these things, and according to the docking results, the interaction observed between these two shows a good degree of inhibition, in indicating that it also elicits some of the therapeutic property against Alzheimer's disease too. But, okay, this data been given, and this data yet to be get published. And of course, I'm stuck over here because of this COVID pandemic. And all this clearly says are supporting, being kept as supporting evidence that our molecule, hydroxycoumarin, has a very good antioxidant activity. And here I'll finish it up, and I take this moment one more time to extend my sincere thanks to Reverend Dr. Alexander and Reverend Dr. Sister Alice, and of course the co-patrons, Reverend Dr. Maria Anthony Raj Father and Maria Orki Raj Father, and my guide, Reverend Dr. Jaya Shanti. And I also extend my sincere thanks to my favorite teacher, Jansi Madam, and Dr. S. Xavier Rajaratnam sir, and the convener, Orki Napoleon sir. Thank you very much, sir. And my special thanks to Dr. Colin Prakash sir. Sir, thank you very much for having me here, sir. Thank you very much to you all.
Thank you, Dr. Jayakumar.
Thank you, ma'am. Special thank you to ma'am. You too, ma'am. I'm sorry. Yes, ma'am.
Yeah. I hope everyone, after hearing Jayakumar's talk, will start including a lot of coumarin containing food in their diet, especially strawberries, apricots, cherries, etc. So that naturally we can, all of us who are diabetic or whatever, we have start consuming a lot of green teas, etc., to provide antioxidant activity. And now that we know coumarin plays a major role in anti-cancer activity, and it's high time that we include strawberries, apricots, cherries that are highly, they contain a lot of coumarins in it. And now it is the time for question answer session. I think two people raised their hand, two participants raised their hand in the Zoom. I think one, Mr. Satish, can you please unmute yourself and ask the question? No, the other person.
Mr. Satish, you can unmute yourself. Ma'am, I think he's not responding. we can
Okay,
go to the
fine.
next one.
There is one more question by Balaji Janardhan, Mr. Balaji.
I read the question. What are the softwares that are available for molecular docking for beginners is the
Yeah.
question. Even I myself is new to this department. As of now, from my student and my colleagues, I learned that Autodock would be somewhat do better. And I have never run the software myself, but still, I heard among just verbal interactions, I would say that Autodock would do better. But anyhow, it is up to you to judicially decide whether you have to go for that software or going to the other one. As of now, I did with my work with Autodock software.
One more question is also there.
Sai Kritika. Thank you, madam. I'll read the question. Is this not possible to produce coumarin hybrids without a tethering agent? Actually, it is a sea of thing. I think I would have shown one example where in which we would have been prepared one hybrid molecule without a tethering agent. But I would suggest, out of my experiences, of course, I'm not exposed to the much level, but still I would say that, just a moment, I'll go into the appropriate slide and I'll come back to you. Emergence, isotin, mechanistic pathway, coumarin fluorescin, in vitro cell lines, coumarin privilege motif. Not this one. My God, just a moment, please excuse me for a minute. Yeah, here you would have been seeing, I think it is better, I think zoom it up. Yeah, I think you can now see, can you able to see this particular one, butyl 2 in coumarin hybrid. Okay, so this is the compound by which without any tethering agent, it has been get added. Of course, but provided it should be have some functional group. So it's better you have some functional group anywhere in the ring where you intend to do the whole. When you intend to do so, by which you can directly put this molecule by with the help of that functional group. I think I'm answered your question. But in my personal opinion, of course, I haven't done much of hybrids, but I'm yet to do. I would say that to start with, it's better have a tethering agent. It is easy to make a tethering agent because that will give added benefit to you. I guess that you can hear me. Can you?
Yes.
Yeah. But this molecule, answering to the question, I would say that this particular molecule being binded with the butyl 2-ene by making a strategy of posting some sort of halogen, substituent here, and it was further being added an agent by which you just this molecule to be get binded in the region. That is the way you can do that. That is one of the way by which you can do that.
Yeah, this is one question from the YouTube chat. Ms. Pungode has asked, how can I separate spots of TLC of SHIP and manage-based derivatives of isotin?
Ma'am, can you just come one more time? I couldn't hear properly, ma'am. Please.
How can I separate spots on TLC of SHIF and manage-based derivatives of isotin?
If I take the question properly, how to separate or purify the isotin molecules, right?
Separate the spots of TLC by SHIF space and manage-based derivatives of isotin.
Well, I'm not very much having sound experiences in isolating isotin. This is what I've been inspired in reading the literature. I yet to do. Therefore, I am unable to. I'm not able to answer your questions rightly, ma'am.
One more question from Dr. Narain.
Yeah.
Docking alone is not sufficient to suggest binding. Have you performed any simulation experiment as well?
No, I told you it is because of the COVID pandemic. I get stuck with this thing. But as of now, I tried. Of course, I am not telling that the docking is the conclusive method, but that is also one of an added supportive evidence. Therefore, I stuck with it. I yet to go on to the simulation studies. If you have any simulation studies, you please let me know so that I myself, you please share your vast knowledge so that I can even aspire for it.
So that's all the questions for now.
Yeah.
Ma'am, Dr. Kesavulu has raised his hand. Can we allow?
Yes, ma'am.
Sir, Dr. Kesavulu?
Yes, sir.
Yes, sir. Please ask. Please ask.
You have isolated some 7 compounds, correct?
Yes.
From Morinda in the ethyl acetate. What
Yes.
is the effect of ethyl acetate other than like a compound will have some effect, but the solvent also will have some effect, correct? What is the effect of ethyl
Of course, actually, of course, I take the question. The point is the ethyl acetate was considered just because of its polarity is concerned. So ethyl acetate, I also undertake a sort of a liquid partition technique by which I added water and other solvents by which I just extracted some of the compound out of my ethyl acetate extract. Therefore, before going into the studies, especially the biological studies, I ensured that it is free from solvent as possible as I could. Therefore, I presume that there is no any hindrance shown by solvent in eliciting the anti-cancer activity. It is also double cross-checked by having the same solvent running in the as a blank in the said biological activity will do the answers. Will give you the precise answer that you really look for.
Sir, I have another question.
Yes.
Sir, actually, you said that hydroxycoumarin is going to bind to monoamino oxidase. Yeah, MAO. So whether it is binding in the substrate binding cleft or anywhere else?
I think I'm new. I told you that I was new to this docking studies. I'll just go by the results that has been given to us, and it says that just a minute. It says that there is sort of an interaction between the protein. I just focusing only the interaction that is happening between the protein molecule, but I really don't know as of now which exact region by which the molecules being get binded. But as of now, looking into the picture, we would draw some, I think you can see the graph. I mean, you can see the picture. The molecule exerts sort of a weak attraction, at least towards the protein molecule. Thereby, we say that this is definitely because of hydrogen bonding was so climbed, and this is the one of the reasons that we found for its anti-cancer activity. Therefore, the study is a sort of a supportive evidence for well-established or otherwise the obtained results. The obtained results are corroborative evidence stays in the caspase test and the gene expression studies. But we also want to see it in a molecular simulation process, especially this docking method, and we find this interesting thing that there is a sort of an attraction. And just because of these attractions, we say that this may also be a reason, or this also a sort of stays as supportive evidence for our claim.
Thank you, sir.
Thank you.
There's one more question on
Yes.
the chat box.
Yes.
Is coumarin can act as antibacterial agent?
Of course, it did. It did, indeed. If at all not coumarin by itself, even I myself did some antibacterial studies, but as far as this presentation is concerned, I considered it was something not pinned to the point, therefore removed. But my answer would be this. Coumarin also shows some antibacterial activity, especially the one which is being halogenated, bearing some electronegative element. The possible reason could be for this antibacterial activity would be because of the electron withdrawing nature, it disrupts the protein that presents in the proteins of your bacterial strains. But of course, it has to be yet. It is up to you to go for a corroborate, validate this information. I read somewhere else. I just struck it in my mind by looking into this question. But of course, the coumarin has antibacterial activity. Even my thesis also been discussing some areas where which the antibacterial activity is being studied.
I think almost all the questions are over.
Yes,
so, thank you, Jai Kumar.
Thank you very much.
In the first session,
Yes.
Mr. Mario had asked one question to Father actually.
Okay.
Why don't we go for synthetic preparation of these compounds? Why we are going for plant extraction? Why we are not doing synthetic preparation when it is the better answer, Father, to supplement with what Father answered? I have an answer to that. The thing is, when we have a synthetic procedure and finding out a particular, preparing a particular compound similar to the compound that is present in nature, we always land up in a racemic mixture. It is always difficult to resolute it into its pure form. Even if you resolute it in its pure form, after some time, it will there is possibility of becoming a racemic mixture again. But in the case of natural products, it will be the optically pure compound. And the optically pure compound has its own beneficial activity on our metabolism. Say, for example, glucose If it is only dextrorotatory, our system will agree to it. If it is levorotatory, it will be fatal to us. It becomes poison to
Yes.
us. So that is a major reason we always go for the extraction of the particularly optically pure compound for its medicinal activity, apart from preparing it in the laboratory. In the laboratory, it is very difficult to separate it into its pure enantiomer. That is to supplement the answer along with my guide, Dr. Ignace Muthu. And now it is time.
Just a minute.
Yeah.
Just a minute. I wish to add on another point along with it. Be it your natural compound or any compound that obtained from a combinatorial chemistry, that is lab chemistry, they do have their own merits and demerits. And as far as pharmacology is concerned, it is more than a chemistry. Therefore, we have to consider the pH, solubility, the mode of administration. For example, we go by intraperitoneal. Yes, ma'am. All these things have to be considered. Yes, ma'am. I finish.
Okay. Now it is time to propose the formal vote of thanks. May I call upon Dr. Sugandha Kumari, Assistant Professor, Department of Chemistry, Auxilium College, to express her words of gratitude to Dr. Jayakumar.
Good afternoon, ma'am. Sugandha,
Over to you, Sugandha.
Yes.
Good afternoon, Sugandha, ma'am.
Good afternoon.
Nice to see you, ma'am.
Good afternoon, Jayakumar. How are you?
I'm fine, ma'am. Thank you very much, ma'am. Happy to see you, ma'am.
Okay. Really, thank you. Really, I'm very happy to propose the vote of thanks today for you. So I deem it a great honor to thank you, Dr. Jayakumar, for your lucid and innovative presentation. Really, it is very useful and informative, and how the drug is designed and the anti-cancer agent should target in multiple pathways is very well explained. Your speech will definitely motivate and encourage the faculty members and the researchers who are all doing on this anti-cancer agents and their biomedical applications. So I thank you for your informative session. I immensely thank the principal, the head of the department, and the organizers of Auxilium College and Sacred Heart College for conducting this webinar. I also thank all the participants for their patient listening. Thank you.
Thank you. Thanks to everybody.
And for the kind reminder of the participants, the feedback link is posted in the chat box. You can just fill the feedback. And once again, I thank you all for joining us today for the sessions, both the sessions, morning as well as the afternoon session. We will log in tomorrow at 10 a.m. with the same link in Zoom for the final session of the 3-day webinar series to be delivered by Dr. Bibula, Head Department of Biochemistry, on the topic hepatoprotective, cardioprotective, and cytotoxic activities of the plant croton passiflora. Happy learning for the rest of the day. Looking forward to meet you all at 10 a.m. tomorrow. Thank you.
Thank you.
Thank you, Jay.
Thank you.
Yes, Dr. Collins, is that?
Yes, ma'am. Thank you so much, Dr. Scholastica, for moderating the whole session so beautifully, and a word of appreciation and gratitude to Dr. Jayakumar, who has given a very beautiful lecture on phytochemistry. So it's bye from both the institutions, Sacred Heart College and Auxilium College for now.